we will talk about a different cell type, which is also located both in the epithelial layer of the lung and of the gut. Mast cells. The mast cells were first described by Paul Ehrlich in his doctoral thesis. Mast cells and basophils share very many similar features. The difference here is that the mast cells are found in the tissue mostly, and very few are in the blood. Basophils can be found in the blood, usually at very low frequency. Less than 1% of all the white blood cells are basophils, in case there is not an ongoing allergic reaction. The basophils may also enter tissue if they are recruited to local inflammation. While the mast cells and the basophils look very similar, they do have different precursor cells. The function of the mast cell is quite confusing. We do not know what the real physiological role is. There are several suggested roles. Often they are built on different types of knockout mice, which also have off-target effects. So the gene that they knocked out is not necessarily completely specific for mast cells. But based on these models, mast cells have been suggested to participate in wound healing, angiogenesis, tolerance, defense against certain pathogens, and in the blood-brain barrier. However, we will talk about mast cells for their role that they play in allergy. The second cell type we will talk about is the eosinophil. The eosinophil can be commonly found in blood. It makes up about 1 to 3% of the total amount of white blood cells. The eosinophil is not very long-lived. It only persists in the circulations for 8 to 12 hours. But once it enters the tissue, it can lie there dormant for quite a while. It has been suggested to be playing a defense against the multicellular parasites, but also in bacterial, viral, and fungal infections. When the eosinophil is activated, and we will come back to how this happens, the eosinophil can undergo cytolysis and throw out its DNA like the neutrophil can do. And instead of netosis, it is called etosis. So while netosis is called netosis because it's neutrophil extracellular traps, the etosis is then eosinophil extracellular traps. They look slightly different from the neutrophil traps. The eosinophil traps are booby-trapped with the granules that are found inside of the eosinophils. And these little granules contain very toxic components, toxic to both cells and to bacteria, which can then be triggered once the net is touching something. First, I will give you an overview of the allergic response. So the first step in the allergic response is that an allergy is stimulating IL-4 producing helper T cells, and the helper T cell will then stimulate the IgE production. It is done by the production of IL-4, IL-5, IL-13 and CD40 ligation to the B cell. The B cell is then class switched into an IgE producing cell. In the tissue, the IgE is sensitizing the mast cell by binding to the Fc receptor on the mast cell. Then, after a second exposure to the allergen binds to the IgE bound to the mast cell. This binding of multivalent allergen will link the Fc receptors with the IgE bound to them, and this will trigger the release of mediators from the mast cell. We will go into the function of the Fc epsilon receptor 1, which is the mast cell receptor. The mediators can be divided up in roughly two categories. It is the lipid mediators and the vasoactive amines, and these will trigger an immediate hypersensitivity reaction, which happens within minutes or even seconds of the re-encountering the allergen. The second type of mediator released from the activated mast cell is cytokines. The cytokines will provide a delayed reaction which can last for hours or for days. What exactly is triggering the first sensitization in an allergic reaction is actually not really known, but the initial response comes from the epithelial cells themselves as a response to an injury or an irritation, and this can maybe be caused by a viral infection in the same time as the allergy is there. The epithelial in the airway will produce IL-25, IL-33 and TSLP, and these cytokines produced by the epithelium will activate the type 2 ILC. And the type 2 ILC will then upregulate GATA3, which is their differentiation marker. The GATA3 ILC in turn will produce IL-5, IL-13 and IL-4. IL-4 and IL-13 will stimulate B cells to produce the antibodies. The IL-13 will also contribute to the process of fibrosis, which is the proliferation of fibroblasts. This is not something you want in the lung, but it may be a consequence of severe allergic and prolonged allergic reactions. IL-5 will stimulate the eosinophils. So once the initiation of an allergic response has happened and IgE is produced, the IgE is driving the allergic reaction. And the IgE drives the allergic reaction by coating the resting mast cells. IgE can coat the mast cell because it is recognized by a receptor on the surface of the mast cell, and this is the Fc epsilon receptor. And the Fc epsilon receptor is constitutively expressed, and the IgE can sit on this receptor for a long time, and without any allergen, nothing happens. However, when an allergen comes around, and in particular a multivalent allergen, an example of this multivalent allergen is the pollen allergen. For example, birch pollen has got several different molecules of the birch allergen, the BETV1, on its surface. So when a mast cell encounters a pollen, it will then lead to a clustering and cross-linking of these IgE-bound Fc epsilon receptor 1s. This clustering leads, as mentioned before, to the release of the cytokines and to the histamines and the lipid mediators. However, the mast cells can also be triggered by other things, not necessarily only by the IgE. Other stimuli could be physical injury and DAMs and PAMPs and various compounds binding G protein coupled receptors. An example of this is morphine, which is binding to a G-coupled receptor, and morphine can trigger an itching reaction, an itching allergic reaction in certain patients. So the biologic effect of the mediators of immediate hypersensitivity is described here in the picture, is the vasodilation, that the blood vessels increase in the area. There's also a vascular leak from these vessels. The bronchi will restrict, and in the gut there will be a hypermobility, which is described as very uncomfortable for persons that have eaten something they are allergic to. Also, a prolonged exposure will cause inflammation and tissue damage. So mast cells and basophils mediator, they include the vasoactive amines, and that is histamine. And the enzymes are stored in granules, as well as our cytokines and the lipid mediators, although the lipid mediators are to a large extent synthesized upon activation. It is these lipid mediators and the vasoactive amines that cause the vascular leakage from the vessels in the allergic reaction. Lipid mediators also contribute to the inflammation, which is the part of the late phase reaction. Enzymes stored in these vesicles are probably also contributing to the tissue damage seen in the prolonged allergic reaction. The effects of eosinophil activation are slightly different because the eosinophils do not contain histamines. The main function of the eosinophils is direct killing of helminths, which is parasites, and also of bacteria. For this purpose, the eosinophil can release peroxidase, which is stored inside of the granule, and lysosomal hydrolases, and also lipases. Another important protein stored in the eosinophil is the major basic protein. The major basic protein constitutes the predominant part of the eosinophil granule, and it is a protein that binds glycosylated proteins. Upon activation, the eosinophil will produce IL-3, IL-5 and GM-CSF. Eosinophils will also produce protein inducing chemotaxis of leukocytes, which means that activation of eosinophils will cause the infiltration of other cells from the blood. The lipid mediators produced by activated eosinophils are mostly leukotrienes, and they cause bronchoconstriction and also increased mucus secretion and the vascular permeability, as mentioned before. Now, this is the last slide, and I would like to mention the immunotherapy against allergy, which is very promising, even though it does not directly involve the mast cells and the eosinophils. So the immunotherapy is giving large doses or increasing doses of the allergen, often subcutaneous or sublingual, also under the tongue, so oral. And these high doses of antigen in the absence of an adjuvant will favor the development of T and B regulatory cells. So the T regulatory cells, you probably know, it's a T helper cell that expresses FOXP3 and the CD25, which is the receptor for IL-2. The Tregs are antigen specific, and upon antigen presentation, and also without an antigen presentation, they will suppress the cytokine production from the T helper 2 cells, which are the driving factor once the allergy is established. The cytokines from the Tregs will also suppress the activation of mast cells and eosinophils, and it will prevent the T helper 2 cells from going into the tissue of inflammation, or where the antigen is. It will also favor the production of IgG4 from B cells and suppress the production of IgE. So IgG4 is an antibody which is only existing in humans, not in mice, and it cannot bind the Fc receptor. So it just coats up the allergen and basically hides the epitopes so that the IgE cannot bind the allergen. B regulatory cells are stimulated in a similar fashion upon large amount of the antigen. The B regs are defined by their production of IL-10, and IL-10 is a cytokine which will affect both the adaptive and the innate immune system, and also suppress the mast cells and the eosinophils. The B reg can also produce the IgG4. So this concludes my lecture. I hope you have enjoyed it. I have taken most of my pictures from either Janeway or from Abbas, and the book here to the right, Cellular and Molecular Immunology, is the book that I refer to when I have the name Abbas in the picture. And if you can get your hands on Abbas, it's a very good book, and especially for this lecture, I recommend you to read chapter 14. So specialized immunity at epithelial barriers, that's called. And also chapter 20 is describing the allergy. this would then cover most of what I've done in this lecture. Now we will talk to each other tonight, and I would like you to write down a question for me on this matter, and then we sit down together and we can use the literature and we can talk about this together. So I see you later, and have a nice day.